Okay, so good afternoon. I got you after the coffee, so I hope you'll be awake. Thank you, Marcia, for the invitation. So my goal for today is first to tell you what are sex chromosome aneuploidies, because not everyone knows, and to convince you that we can use sex chromosome aneuploidies to think about and understand better sex differences in psychiatry. I hope that by the end of this talk you will be as enthusiastic as me about this subject, and also will consider to team up with us if you have complementary expertise to ours. So I'll motivate this talk by this quote that says, it is not the act of justice, but of foolish injustice to pretend that the sexes are the same. Justice is exercised in respectfully providing for the due needs of each. And I would say that medicine is exercised by respectfully providing for the due needs of each. And in this era of precision medicine, when we know more and more about the genes, I would claim that sex chromosomes are the basic biological point where this differ, that the sexes differ, and we can start thinking about sex differences. So today I'm going to present you the sex dimorphism in mental disorders. I'm going to talk about sex chromosome aneuploidies. Then we will examine by data and see if sex chromosome aneuploidies can serve as a model for psychiatric conditions. And I will review some insights from the study of sex chromosome aneuploidies. I'm going to use a review paper that we published this year, and again, we'll ask you to team up with us. So, as mentioned before, and this is just more detailed and visual diagram, we know that sex differences are the rule in psychiatric conditions, and we do know that neurodevelopmental disorders such as intellectual disabilities, autism, and ADHD are much more prevalent in the males than in the females. The opposite is happening when we start to look on mood disorders and on anxiety, where we see that females are much more suffer much more from these conditions. There's also, it's not only in the prevalence, it's also these sex differences also affect our management, and the way it affects our management, there's many studies that been showing that. And just as a sample, in ADHD we know that their females will respond differently to methylphenidate, the stimulant that we give for ADHD, than males. They will have a better response in an hour and a half after you give the medication, and a worse response at the end of the time of the medication, that the medication works. We know that there is a sex differences in the response to antidepressant. Male will respond better to tricyclics, while females will respond better to SSRIs, but this disappears with menopause. We know that schizophrenia, and it's been discussed before, schizophrenia is not necessarily different between the sexes, but the disease looks different. It will start early in the male. It will be the course of the disease probably will be more severe, and females also have a better response to antipsychotic medication. They will show a better response to antipsychotic medications, but they will also show more side effects. For example, hyperprolactinemia that we see will happen more in females, but even if you correct for the higher prolactin to start with. So there are challenges, and if you think about how complex is our genome, and the fact that we still define psychiatric disorders by behavior, this complexity together makes it very, very difficult to think what is the contribution of the X chromosome, what is the contribution of the Y chromosome. And what I'm going to suggest today is that the sex chromosome aneuploidies is actually a condition in nature that we have variation in the number of X chromosome, in the number of Y chromosomes, and this is a window for us to understand what happened to the brain when we change the number of the X chromosome, what happened to behavior when we change the number of Y chromosome. So what are sex chromosome aneuploidies? And there's more than this table contains, but the ones that I'm going to talk about today are the ones that are we reviewed in a review that you see, and they are the most common. So we have Turner syndrome that's caused by X0, meaning that those females, as Marcia, I think you mentioned that all of us have an X chromosome, and it's correct. Males and females have one X chromosome. These females have only one X chromosome and not the second one. Those girls will present with short stature, and the most important thing, and I think, and we're going to talk about it a bit more, is that they also have premature ovarian failure, so meaning they're not only have a deficit of the X chromosome, they also have a deficit, hormonal deficit. The second syndrome I'm going to talk about is Klinefelter syndrome. And these are boys who have extra X chromosome, and they will be tall, and some of them will also present with fertility issues. And the last two syndromes are easy to remember because they're by the sex chromosome aberration. So it's XYY syndrome and XXX syndrome. And what I want to note about those, there are some possible phenotypes, but also many of them are going under our radar because these individuals are fertile. So, and they're more or less with the standard height. So we're not sure how many of them there actually are. And a bit into the biology of what happens to the genes on the X chromosome, and I will focus specifically on Turner syndrome because that is where my research and the researcher will present you now is. I want you to focus now here. So this is a typical female, okay, XX. What happens in the body is that one of the X's goes in activation, and there's several genes that escape this inactivation. So basically, while the male has one X chromosome, the female will have also one X chromosome, but an extra copy of several genes. Okay. What happens in Turner syndrome is that we basically missing this whole second gene, and we don't have the haploinsufficient to these green genes. However, recently we saw something that emphasized the importance of transcriptomes, and not only looking on the genes, is that we saw that actually Raznan et al. Showed that for girls with Turner syndrome, they, as expected, going to be deficient. They won't have those genes, this gene expertise. I can say that now, but they will have overexpression of other genes that we did not expect. So it's not only if you have the gene or do not have the gene, it's also what happens to the regulation of the gene on the X chromosome. And so first of all, before you even need to listen to me, I need to prove you that sex chromosome aneuploidies actually can represent and manifest psychiatric condition the same way other conditions happen. And this study I did that was the purpose of this study. And we did it in girls with Turner syndrome. It's very important to emphasize that girls with Turner syndrome have normal general intelligence, which means that we will not not attributing their deficits in executive functioning, and if they present with ADHD, to their deficits in IQ. What we did is we took a group of girls with Turner syndrome, we compared them to typically developing controls, and we compared them also to idiopathic ADHD, meaning children with ADHD without a genetic condition. And what we saw, and we measured them on measures of both behavior, ADHD behavior, and also measures cognitive behavior of executive function that are associated with ADHD. And what we were able to show is that 50% of the girls with Turner syndrome presented with some higher level of symptoms of ADHD. 25% of them had fulfilled the clinical criteria for ADHD. And the girls with Turner syndrome was as inattentive and hyperactive as boys with ADHD. And they were more hyperactive than girls with ADHD, which might make us think that the fact that the single X chromosome in Turner syndrome and the single X chromosome in boys is connected. And we saw the same thing in our cognitive data of executive function and attention. And you see here, these are the children with typically developing children with performance on the hundreds, and girls with Turner syndrome and children with idiopathic ADHD were equally lower on these aspects. And we then move forward to look also on the brains of girls with Turner syndrome. And here I just want to set the stage with very, very brief introduction to the method. So we used resting state functional MRI measures. And what this shows, it's one of the first studies that used resting state. So basically, think about a person that lies in the scanner. He gets a motor task to tap with his finger, and the motor network lights up. But when this person, the same person, rested, and we put a dot here, or in the, where is the letter A, and we basically had a sphere around this dot, and we looked on which part of the brain are correlating with this dot A. We saw that the same circuit lights up. And we think about it as if it works together, it fires together. And this can be expanded, and based on where you put the seed, you can actually identify those networks, different networks. And the networks that we focused on were attention networks, and I'm going to talk about two. One of them is the orientation and orienting network, which basically means that if I put my finger there, some of you might look there, so you're orienting your attention to the entrance of the room. So we have a dorsal attention and a ventral attention network. And we also have the executive control, meaning this network basically responsible on you guys sitting here and listening to me, even though that maybe you want to go and do other things, but you kind of like can suppress those urges. And what we did is basically we took girls with Turner syndrome and compared them to controls in the scanner. And what we saw is that only for girls with Turner syndrome, there's hypoconnectivity on the dorsal attention network and in the frontal parietal control. And this is just an example. What I want you to appreciate here is how much these networks are overlapping. So if you measure a group of kids with ADHD that are very, very heterogenic in the terms of genetics, you have a huge difficulty to identify which networks are affected. But in Turner syndrome, it's easier. We saw the same results of hypoconnectivity with a different type of analysis, but we also saw that there's, in a different type of analysis, when we only focused on the parietal lobe, we also saw that there's hyperconnectivity between the parietal lobe and the cerebellum. And when we correlated that with behavior, with the measure of executive function, we saw that only for the girls with Turner syndrome, there's a positive correlation between this hyperconnectivity between the cerebellum and the parietal cortex and the performance, meaning that we might looking at compensatory mechanism. So in conclusion, this is an example how sex chromosome aneuploidies can help us dissociate between attention network and connect that better to the genetic aberration. In this case, it's the 1X chromosome, and also identify some compensatory mechanism that might be actually applicable clinically for the girls. And now I'm gonna switch to more general insights that we had from the study of sex chromosome aneuploidies, and I'm gonna go over these 6 insights. So if we look on all the syndromes that we reviewed and studied, so we do see that overall a change in sex chromosome number affects behavioral phenotype, okay? So it's across the board. It's for ASD, for ADHD, depression, and anxiety. There is very limited data about the XYY and XXX, so I'm going to be more careful when I'm looking for that. And also, I think this limited data also expand this very high percentage of ADHD and ASD that are probably lower in reality. Another insight, and I do want to orient you for this graph. What we see here is the rates between the rate between females and males for ASD. It's 1 to 5. When you compare, then what we did is we compared the rates that we found in the literature or from our studies, and we compare each one of the syndromes to either XY males or XX females. And we were able to show, and you can think about it as a simple subtraction of XYY, for example, minus XY. So it gives you an extra Y effect. And we saw in general that there's the effect of the extra Y or missing 1 X chromosome has a huge effect on the prevalence of ASD and sex chromosome aneuploidies. We also saw an overall increase in ADHD for all sex chromosome aneuploidies. And again, this finding is I'm kind of like stay carefully because the data is, I don't think it's good enough. And this is something that we're just now working on, and I want to emphasize more in depth some things that insight that we might understand that we cannot reach otherwise, is that what we did here is we actually took a group of kids with Turner syndrome, girls with Turner syndrome, and boys with Klinefelter. Up until now, most of the literature compare girls with Turner syndrome to girls and boys with Klinefelter to boys. On these cognitive measures and behavioral measure, we felt comfortable to compare them also to the opposite sex. And just to remind you that all these measures are corrected for sex and age, okay? So what you can see here is that, and down there is effect sizes. So 2 things I want you to kind of like take out from that. So we do see that for some measures like shifting or auditory attention or sequence number, this is a measure of it's a measure from the IQ testing, a measure of working memory. we do see that there's no difference if you compare a Klinefelter kid to XX or to XY. Okay, so, and there's no difference if you compare a girl with Turner syndrome to X0 and XX, meaning that it's the aberration that the number of the sex chromosomes that affects that. It doesn't matter. The delta is not different if the delta is compared with an X or an Y, or Y. I hope, is it clear? It's like, it's very, so please ask me if it's not. But for specific measures of processing speed and working memory and hyperactivity, we see something different. We see that if you're missing an X chromosome, the effect size, the actual clinical effect on this measure is very high. If you add in an X chromosome, there's no effect. This is the fact that it crosses the 0 means that it's not significant. And if you either have an X or Y or minus Y, it doesn't matter, it's the same. So we think that we can understand a little bit more about the fact that the X chromosome might confer in extra, it might confirm a protective effect, while missing an X chromosome is very, very significant to the development. So we do claim that the direct comparison between same-sex but also opposite sex might give us insight about psychiatric conditions. And okay, so and this is about mood and I have to say that the data on mood is much more limited, but I'm going to use that as a segue to discuss something else. So we looked on depression, and we see that there's depression also in the Turner syndrome, in Klinefelter syndrome. There's some reports that even up to 70%. And again, there's limited data in the other section. It might be there are some limited results that that the extra X chromosome in Klinefelter and in the extra X chromosome might confer a risk to depression in Klinefelter. It might be. There's also high prevalence of depression in Turner syndrome, and it might be associated with the lack of the X chromosome. But just to remind you, those girls are also hormonally deficient, and they need exogenous hormones to replace the missing estrogen that also might play a role in mood. On top of that, those kids are suffering from different development. They're shorter than other kids, and there is a delay in sexual development that might also play a role in depression. But depression is also, the segue I'm going to use that is to discuss a little bit about the immune system. So the immune system has been increasingly spoken and discussed also by Daniel that gave the talk earlier, and as a risk for schizophrenia and mood disorders. And what I want to show you now is that the data we collected is interesting in that way of autoimmunity, and to see how that might be connected, how the mood disorders, the autoimmunity, and the sex, the number of sex chromosomes might interplay. So what you see here is the prevalence of autoimmune disorders in Klinefelter syndrome. It's very simple. In orange, you see that the diseases that are typically female predominant happen more in females, and in green, the ones that happen more in males. And you see that in Klinefelter syndrome, there are high prevalence of female predominant autoimmune disorders, one of the most significant, one of the most prevalent one is SLE, which we know that is associated with very, very, not severe, but it is associated with psychiatric conditions such as depression and also others. And the opposite, semi-opposite, it's a more complex figure here, picture here, is that for Turner syndrome, one thing we see more of male predominant condition like the ones on top, but we also see a lot of autoimmune diseases, which generally, if we see more of them in Klinefelter, I mean, it's the X-chromosome, we're not supposed to see them in Turner syndrome. But I do want to point your attention that it's very specific autoimmune disorder, many of them related to the thyroid. So it might be a more specific risk in this population. And the last thing I'm going to touch base on is it's a survey that we did on the GWAS, because we were interested to see, okay, we see more psychopathology in sex chromosome aneuploidies, so we wanted to see if that psychopathology comes up on the X chromosome from GWAS studies. And we surveyed the GWAS catalog, and we found 86 SNPs that associated with ASD, and 26 SNPs that are associated with ADHD, and none of them was located on the X or the Y chromosome. And we kind of like, it was very puzzling, so we actually checked how many of them reported the X chromosome or the Y chromosome, and 54% of the studies of ASD included or reported the X chromosome, and 15% of the ADHD studies. So not necessarily it's not there, we might just not look at that. And this is kind of like a call to say that we need to include the sex chromosome in our GWAS studies, and there are many technical limitations to that. But we also, and this is done in medicine in other diseases like Crohn's disease, is that people go back to the data and just look at it now. We might find new things. And the clinical implications, so just remember the sex chromosome aneuploidies are common, they're 1 to 400. If you see them as a clinician, just be aware in childhood to think about attention problem, visual spatial weaknesses, language development, and social communication, and in young adults for depression and anxiety. We invite you to team up with us if you have complementary expertise to ours. And I just want to thank 3 people. First to Alan Rees, who was my PI on most of this work. Today I'm working on also on other things. So a lot of what you see is data that were collected under his grants. To Shira Flash, Dr. Shira Flash is here and wrote with me the review. And just to present David Hong, that will talk after me, and he gonna give the hormonal aspects and talk about the transgender brain. And thank you to Wisdom and MCHRI. I had to remember the M for funding, and that's all.